This video is about molecular events in the cell cycle. We're going to use three different proteins in order to illustrate how the molecular events of the cell cycle are controlled. One from early in the cell cycle, during prophase, in which histone proteins, shown here with DNA wrapped around them, need to be produced in order to allow DNA to wrap around and form tighter and tighter, more condensed strands of genetic material. Then, midway through mitosis, to make microtubules, tubulin is necessary in order for the sister chromatids to line up on the metaphase plate and be pulled apart later during anaphase. Finally, in telophase, actin is needed to help form this cleavage furrow that in an animal cell is going to divide a single cell with two nuclei into two daughter cells. So the cell cycle occurs over time, and I'm going to plot on the y-axis here the amount of protein for particular genes during the M phase mitosis, G1, growth 1, DNA synthesis, G and G2. We need an increase in histone toward the end of the S phase, as DNA has been made and now needs to be condensed. In G2, we need an increase in tubulin because we're going to need mitotic spindles and just increasing sizes of the cell, and during mitosis we'll need more actin. So here are the genes: actin, tubulin, and histone. We need messenger RNA from each of these in order to make the proteins. We need a transcription factor to bind. Transcription factor that's been phosphorylated needs to bind to a particular sequence, and I've written some G's and A's and C's and T's paired up, where our transcription factor A will bind. That phosphorylated transcription factor then allows RNA polymerase to bind at the beginning of that gene and move along the gene and make messenger RNA for histone. Well, how did we get that phosphorylated transcription factor? A kinase, we'll call it kinase 1, is phosphorylated. That activates it. It holds an ATP in a binding pocket. It binds to TFA. And transcription factor A winds up with a phosphate from that ATP, and an ADP is lost. The kinase, kinase 1, can go back and repeat with a new transcription factor A. How did we activate kinase 1? For that, we used a cyclin. The cyclin was bound to a cyclin-dependent kinase that has a binding pocket for an ATP. That cyclin-dependent kinase bound to the inactive kinase 1 and was phosphorylated. Second step, third step, we got TFA, fourth step, TFA binding, fifth step, RNA polymerase, and sixth step, messenger RNA from histone. We repeat this for the other genes, each with a different cyclin and CDK partner. So I'll call it cyclin 2, they're actually lettered. But here's a partner, CDK, for cyclin 2. We have another kinase, I'll call it kinase 2. There's the phosphate added to kinase 2. We have activated kinase 2. So it can now put a phosphate on another transcription factor that I'll call B. There's our phosphate. Transcription factor B changes shape. It'll now bind to a particular sequence of DNA bases. Once phosphorylated, there's our specific bases. And that allows RNA polymerase to bind, fifth step, move along the gene, and produce messenger RNA for tubulin. For actin, we repeat. Later, another cyclin that I'll call cyclin 3 activates yet another transcription factor through another kinase. Transcription factor C recruits RNA polymerase, moves along the gene, and we get messenger RNA for actin. So going back to our different phases, we have mitosis, G1, S, G2. One cyclin, I'm calling it cyclin 1, goes up. Cyclin 2 peaks at cyclin 3, the transition between S and G2, and cyclin 4 in the transition between G2 and M. The actual letters are D, E, A, and B for these four cyclins, but I'll just call them 1, 2, 3, 4. I'm not concerned that you remember the names. Again, there's a particular sequence on the DNA to which a transcription factor can bind. And I called this transcription factor A, that's active when it's phosphorylated. We needed a cyclin, in this case cyclin 2 for histone, in order to phosphorylate a kinase. That phosphorylated kinase then phosphorylates transcription factor A, allowing transcription factor A in this new conformation to bind to DNA. And here I'm drawing histone. Since cyclin 2 is between G1 and S, we might actually expect a different gene. But we get messenger RNA for that gene. And we repeat, for any gene that needs to be activated at a specific time, different cyclins can activate that gene. So we repeat for tubulin and actin. This represents a hypothesis and a model for how cyclins might regulate gene expression and thereby regulate the events of cell reproduction.